Well, good morning, everybody. As I said just a while ago, my name's Matt Paratt. I work for Forest Research and normally deal with trees and have a special interest in conifers. And if you're very lucky, you could get to come on one of the conifer ID courses I run for BSBE and others. But today it's all about grasses. And here's our program. So we're going to have a bit of an introduction to grasses, during which I'm going to go through the naming of parts and things and get you all up to speed with the terminology, really. And then we'll have a short break with a little bit of a task for you to do during that break. So let's get on. So firstly, what is a grass? They're flowering plants in the Poaceae family. So this is the grass family. They're monocots with simple leaves with parallel veins. Now, a monocot refers to the number of first leaves that come out of the seedling. So in a dicot, which is the majority of the flowering plants, you have 2 seed leaves, and in this case, just the one. So a single shoot emerges. Key feature is that they have hollow stems, which are called culms, and nodes where the leaves emerge. Now, the nodes look like little knobbly knees, if you like, but we're going to visit that shortly. Critically, they're wind pollinated, so insects don't have a part to play in the pollination of grasses. It's all down to the wind. And if any of you suffer from hay fever, you're probably all too familiar with their vast amounts of pollen that get shunted around in the air around this time of year. So the other really important character is that the new growth in grasses starts from the base of the plant rather than the tips of the shoots. And this is a superb adaptation to grazing. And if you've seen the David Attenborough films with wildebeest sweeping majestically over the plains, to quote Monty Python, then that gives you a very good idea of where grasses are really at their best, because they can be grazed and grazed, and the growing point is never cropped out, so they can still throw up new growth. But are they important? Yeah, very. Economically, extremely to us as a species. So 70% of all of our crops are grasses. And amazingly, just 3 of those, rice, wheat, and maize, provide more than half of the calories that are generally eaten by human beings. So this family, the Poaceae, is probably our most important plant family that we consume. We've also got other grains like rye, barley, millet, oats, and sugarcane, which is also a grass. Now, I'm not sure how much of our sugar still comes from sugarcane, but it's a source and it is a grass. So they are also essential ingredients for that fun thing, alcohol, and especially beer, whiskey, and vodka. Their base is all grain, so all grasses. These are all quite heavily bred grasses, quite different from the wild forms, but they're still grasses. And that character of them being supremely suited to grazing means that they provide a lot of fodder for our livestock. Not only the fresh green leaves, but also the grain. So an awful lot of the grain we grow goes directly to animal feed. It bypasses us completely. And building materials as well. So things like thatch in the south of England especially, and bamboo, if you're in the Southeast Asian region. Bamboo is a fantastic scaffolding material as well. And forget metal poles, it's all about bamboo. So it's a big, big family of plants. So there are about 12,000 species, give or take, worldwide. And it's the 5th largest plant family. And I think Jim and I are both amazed to find that it is actually behind a family called the Rubiaceae, which includes things like cleavers, Galliomaparinae. So it's a big family, but not the biggest. And there are about 100 grass species which are regarded as native in Scotland. And that's a huge number, but it's not so big as the number of non-native grasses that are all over the place. Now, these tend to be short-lived. And one important thing to say about this course is we don't cover all the grasses you might come across. We're covering the really common things that you could see just about anywhere, outside your back door, along the street, in the park. So there are a lot more to get to grips with. The grasses also form a really important part of habitat. And grasslands cover something in the order of 70% of the Earth's landmass. And they are present on every single continent. So key skill, is it a grass? or something else. And I'm going to dive into a subject which sometimes panics people here, common names versus scientific names. So this plant on the right is a cotton grass, but it's not technically a grass. And one of the key skills you're going to be able to pick up today is how do we tell the difference between the two? So this picture is lifted directly from the handbook, which I'm sure you all spent last night browsing thoroughly. Maybe you didn't, Jim, actually. So Jim was entertaining friends last night. So, but this is taken directly from the book. So if you could now find your bag, which is labeled A, B, and C. So it's just got 3 things in it. So does everybody have A, B, and C to hand? Yep, that looks good, Deborah, in a nice glass jar there. Okay, so let's have a look at sample A. Does everybody have that to hand? Great. One of the real skills with plant identification is before you even look at the words in your guidebook, have a look at the plant. Just get familiar with it and try and pick out any key features. So the first one I'd say is, and this doesn't always apply, but it's a good starter, is if you hold the stem between your fingers and try and roll it between the fingers, a bit like this. How does that feel? So we're looking to decide, is it nice and cylindrical, rounded? Is it flattened, or does it have some really distinctive edges? So would anybody like to venture, click on their unmute and venture whether that is a rounded or does it have distinctive edges?
Distinctive edges.
Yeah, excellent. So if you have a pair of scissors nearby, chop through that and hopefully you can have a quick look and see what the cross section looks like. And it should look a bit like a Toblerone bar. So nice and triangular. And so this isn't always the case, but it's a pretty good rule of thumb. If you've got that nice triangular stem, looking at that diagram up on the screen, which one do we think we're looking at there?
Sedge.
Yeah, thanks, Alison. It is a sedge indeed. So a couple of other characters about the sedge. The leaves come out of the stems in a set of 3. So if you think of a spiral staircase, the best way to see this is if you actually look down the plant or up it, in this case, the leaves do come out in what we call 3 ranks. And that reflects nicely in this triangular stem, actually. And finally, the business end. Now, these aren't fantastic at the moment because it's been a very late spring, so things are just emerging. But right up at the top of the plant here, this is the inflorescence. So these are the flowers. And in the sedges, the male and the female flowers are separated. So it's very difficult to see at the moment. But right at the very top here, we'll have the male flowers that will produce anthers. And then lower down are the female flowers that will eventually have the little seeds. Okay, great, so that's a sedge. So let's pick up sample B. It'll look a bit like this. And again, a good first test, just roll it between finger and thumb. Does this one have edges?
No.
No, great.
No.
So it's quite rounded. Yep. And what I'd like you to do is take that shoot. If you've got your scissors handy, chop it. But if not, it should tear quite readily. And just have a look at the cut end where you've torn it. And we're looking to see if it's hollow. So how's that looking to everybody? Is it hollow or is there something in there?
It's not hollow.
Yeah, great. Now, with a bit of careful peeling of this outer green layer, you can probably reveal this very white, pithy pith inside. And this used to be used for lamp wicks, incidentally, because it's really good at conducting fluid. So looking back at our diagram, which one of those do we think this might be?
A rush?
Yeah, it's a rush. Now, we haven't got flowers on most of these samples, again, because it's quite a late season. But if it did have flowers, they actually do look like little tiny, sort of traditional six-tepaled flowers, little stars, really. They're quite pretty, and it's worth using your lens to have a look at them. So that's a rush. So you can probably deduce what the final sample is, but let's have a look at it. And just in case, we're going to look at all of these bits more closely shortly. So this is quite a huge sample, and in lots of cases, these have been bent into all sorts of shapes to get them into the bags. But for my money, the key feature to look for, if you look along the stem, you'll eventually come to what look like knees. They're little knobbles on the stem. You might have to hunt for them a bit. They may be covered up by leaves. But this is a node. Now, these are absent in this form in the others. So if we start to see these thickened, knobbly knees of nodes, we're thinking grass, really. And we'll just go through and look at the other features in a bit more detail. Okay, has everybody found those nodes? Yeah, I can see a few nods, great, good. So remember those three. Now there are a few others. So there are some true flowering plants or forbs, which can look a bit grassy. I haven't put any of those in because they tend to wilt a bit too quickly, but be aware of those, and also wood rushes. But we won't worry about those today. A grass, a sedge, and a rush, they're the three that you could have most trouble with. Now in the book, there is a little key to help you go through that, but we're going to move on to grasses proper. So botanical terminology can be quite off-putting. It's a bit of a dark art, it's a second language, but it can really help you because the words describe specific things on the plants. So let's have a look at them. So going back to your grass, we've talked about nodes, these knobbly structures. Now the bit between two nodes is a culm. So a culm is just a stem. So if you can find two nodes, and the easiest way to do this with this big specimen is to run your fingers along it, and you'll find the big obvious ones, but some of them might be hidden behind some other material. So those are a pair of nodes. Now, a node is where a leaf actually starts on a plant. Other plants have nodes, but they're not as big and obvious as this. So nodes and culms. Now, bamboos are grasses, and they're a fantastic place to see nodes and culms. They're blatantly obvious. So if you've got some bamboo locally, go and have a look at those. It's kind of grass in giant size. So leaf blade and sheath. Now, these things are devilishly difficult to photograph, partly because they're so big, but also so wispy. So on the grass, we've got our stem, our culm, and we've got those nodes, but then we've also got leaves. Now the leaf blade is this flattened piece up here. And if you follow that down to where it joins the stem, if you slightly start peeling away from the culm itself, what you've got there is the leaf sheath. So the leaf sheath wraps around the culm below it. Now, some of these can have features like they can be roughened because they've got little teeth, or they can be slightly inflated. And those sort of characters can really help you pin down which group of grasses you're going to be looking at. So leaf blade and leaf sheath below it, okay? Ligules, this is the really fun bit, looking for ligules. So going back to our leaf blade, if you take your leaf blade and follow it down to the stem again, and just pull it very slightly away, and if you like, tear it off below that point. Now, if you wrap it around a finger like this, so hold it between finger and thumb and actually wrap it around, you can then pinch that in place so you can get a really good look at it. So hold it with the blade facing away from you, wrap it around your finger. And at the point where that leaf blade joined the stem, I'm hoping you'll all be able to see something that looks a bit scruffy, in this case, slightly brown and papery. Is everybody managing to see that? So it'll look, I hope, like the image on the left of the screen. And this is a ligule. Someone usually at this point says, what does a ligule do? And I don't have an answer for you. It just is. Okay, but they're very useful in helping us to identify the grass because they come in different forms and different shapes. So some grasses have a ring of hairs rather than this sort of papery tissue-like ligule. But all the ones you're going to see today have this papery membrane. And it can be different shapes and sizes. So looking at the pictures on the screen, we've got in the middle a very short ligule, a bit like a collar with a slightly corrugated upper edge. And on the far right is a ligule that's long and pointed. So it's another good feature to have a look for when you're first faced with an unknown grass. So ligules. The main thrust today really is all about the flowers or the panicle or the spike. We've got two main types, panicle and spike. Panicles you can think of as looking like Christmas trees, in essence, once all the needles have dropped off. So we've got two main types of panicle really. We've got the one on the left, which is irregular. So the separate little stalks within that panicle come out at different points. Or in the middle of this picture, we've got a whorled panicle where they all originate at the same point on the stem. So looking at your grass, which one of the two is it? Is it irregular or whorled?
I think irregular. Irregular,
yeah, good. And one word of warning with panicles, and this is something we encountered as collectors earlier this week when we were out looking. The picture on the right is a panicle which isn't yet fully emerged from its leaf sheath. And it can look quite different. So be cautious when you find a grass as to whether it is fully emerged and you're getting a true idea of the shape or not. So since we collected earlier in the week, things have really moved on and we've now got all sorts of things which are now looking good, unfortunately. But it does mean when you go out to look around, you'll see some really good examples. So the other type is spike. Now, spikes, again, come in a couple of different flavors, a couple of different shapes, and you'll encounter all three of these ones shortly when you start looking at individual species. So they can be pretty scruffy and irregular on the left. They can be nicely cylindrical in the middle, and they can also be one-sided in one particular species case, and that's a really good clue as to which one you're looking at. You might see some other things on these pictures, little sort of scruffy bits and pieces stuck on the outside of the spike, the inflorescence. And these are the actual flowers, because remember, grasses are flowering plants and they have real flowers. They're just not quite what we're used to when we think of things like buttercups. So this is a really important bit of the structure. It's difficult to see. Did everybody manage to get a hand lens? Because they can be your best friend as a botanist, and they don't need to be expensive. I see everybody looping them around their necks as we speak. So little hand lens, and these are essential really for seeing some of these smaller features, unless you're blessed with fantastically good close-up eyesight. Quick tip on how to use a hand lens. Don't hold your hand lens down to the specimen, miles away from your face. Put the lens up to your eye, and then carefully, so you don't poke yourself in the eye, move the specimen up until things come into focus. And that way you'll get a much clearer image of what you're looking at. Okay, so that's just a quick hand lens tip. Times 10 is good enough for most of this. So what we're looking at in the flowers are these clusters called spikelets. And these are, if you like, the smallest unit that you're going to find on your little flower. And we want to look to see whether there's a single floret in there or multiple florets. Now, each floret is made up of male and female bits and a couple of leafy sort of membranous bits called the lemma and palea. Quite often, and I'm just going to go back a slide. So in this middle picture, we can see some sort of purpley browny bits poking out. So those are the anthers or the stamens, the male bit of the grass. Now tucked in beside those, you'll find little feathery bits. Now these aren't visible until the flowers fully open up. And that needs a nice warm, preferably a dry, still day. If some of you had these on your windowsill, some of your flowers may have opened up, but I suspect a lot of them haven't. But we'll be looking at those in a bit more detail as we go through the specimens individually a bit later on. But don't worry too much about finding out how many florets are in each spikelet, but be aware that it's worth having a little look. It can be helpful. One other important character, which isn't actually mentioned in the book it's directly, is before you even start picking a small piece of your grass, have a look at how it's growing. So is it a clump, a tussock, or a dense tuft, all originating in one spot? Or does it appear to be widespread? So the picture on the left is an example of a tussock forming grass. So that tends to stay as a tussock. It doesn't spread vegetatively. It just spreads itself around by seeds. Whereas other grasses produce these structures which enable them to cover ground. Does anybody here grow strawberries? Yeah? So you're familiar with runners which shoot off from the strawberries. That's a vegetative means of spreading around. Now, we've got two types of structure. One of them is above ground and the other is underground. The above ground one, it's a modified stem. It's called a stolon. So if you grow strawberries, those runners you're seeing are stolons. How about couch grass, the sort of nemesis of allotmenters everywhere? If you're familiar with couch grass and the way it spreads underground, those are rhizomes. A good way of remembering it is rhizomes are like roots but under the ground. So that tells you the difference. It's worth having a look for those with some of the species that you might see. And you might have to pull them out of the ground a little bit, or you may see them sprawling across the surface of the ground. So rhizomes and stolons. And that relates directly to whether things are clumpers or spreaders. If it's a clumper, it's staying in one spot. Mm-hmm. It's not moving around. If it's a spreader, then it needs to be able to move out from that clump. Okay. So when you start to look around, you'll see that it's not got this densely tufted appearance. It'll form a creeping mat. And so those are the spreaders. Okay, right. So the ones that are in clumps don't have either of them? No.
Well, not quite. The ones that are in clumps can have very short rhizomes. So it's all to do with the length. Nearly all grasses do have rhizomes, but it's all to do with the length of the rhizomes. So the ones which are form tussocks or clumps have very, very short rhizomes. The ones that form big patches over big areas, like Yorkshire fog, they do have rhizomes and they're very long.
Yeah, in the clumpers, you won't see them. If you go excavating around, they won't be obvious.
Okay,
thanks. Any other questions?
Yes, I think I might be on Zoom. What's the palea?
Oh. Thank you, Jane.
I couldn't join chat, so I thought I'd just jump in.
Yep, no worries. So the floret structure is the very smallest unit. So these are the two membranes, if you like, which surround the actual flower itself. So the stigma, the ovary, and the anthers. And they're just like two modified leaves. The easiest way to remember is the lemma is the lower one, and the palea is the upper one. When the seed forms, they enclose it and wrap around it.
Thank you.
Okay, no worries.
So this was the last one that we were doing. We were counting florets per spikelet, which is always the tricky part. So here we go. This is an example of 3 florets per spikelet. You can see the glooms, a pair of glooms at the bottom, and you can tell there are glooms because they're in a pair at the bottom where the spikelet joins the stalk, and 1, 2, 3 florets. So counting florets per spikelet, I think, is what people usually find the trickiest part, especially if there's only 1 or 2. We, on our sweet vernal grass, had a, had a bit of an issue finding both awns. It took a little bit of dissection and pulling it all out, but again, I can't move my mouse. You can see the little membranous gloom at the bottom, and then we've got one long, quite well developed floret with its awn, and one awn that was slightly closed inside the spikelet, but when we pulled it apart, it was still there. And we also, while we were on crested dog's tail, we thought we'd use it, this picture, as an example of the difference between sterile and fertile florets, because crested dog's tail has an entire sterile spikelet. That's the one on the top of the picture, clasped in a pair with the fertile spikelet. So the fertile florets are the big fat ones, and the sterile florets are the little skinny ones. And you can see there's one spikelet that's all sterile and one spikelet that's got 2 fertile, possibly more in there. I can only see 2 in the picture. But yeah, I just took those pictures because they were quite nice things to see big. The only hard thing about grasses is how small some of the pieces are.
Okay, so we've dipped our toes into the grass ID, but where'd you go next? Providing you're still interested enough to pursue it, that is, we haven't put you off completely staring at florets and trying to decide how many of them there are in a spikelet. But here are some good steps to get you further along your grass identification journey. So, books. There are plenty of useful books out there. Here are a few of the ones that I think are worth a look. There are plenty more. So, not a book to start with, but the Field Studies Council produces these fold-out guides, which are an excellent introduction to various plant groups, animals, insects, but they're one on grasses, the same as this course really looks at the common grasses. So you get some good illustrations, a glossary, and also a good key as well with a few other facts and things. The great thing about these is they're only about £3 or £4 each. They're laminated and they slip into a rucksack very nicely. So they're worth a look. Stepping up the game a bit, we wander into Hubbard territory, and this is the classic grass ID textbook. It's quite a few years old now, but still widely used. The one thing about this one is the terminology, the taxonomy has changed a little bit. So things have been updated and the old names still in the book. So you might have to go through and change a few of the names, but the common names haven't changed. They're still the same. Hubbard is good. Illustrations are excellent. Get really serious, you can move into Cope and Gray territory. Personally, I find this one much more difficult in terms of identification, but I really like the illustrations and I really like the descriptions that go with the text. So I'll tend to use something else to do my identification, but then I'll come back to Cope and Gray to actually read a bit more about the grasses in question. And finally, if you haven't got inflorescences, so you can ignore spikelets and how many florets you've got per spikelet and dive straight into vegetative ID, I'd recommend having a go with this because it works surprisingly well with grasses. The key isn't always easy to follow because of the way it's laid out, but persist. It's got some really good, useful tips in it. I should say as well, we're going to send you out a Taking Things Further sheet after today's course, and all of this will be included in that. So don't worry about scribbling things down. And we'll include links to useful websites and things as well. Bit of a plug here, if you're not already, join the BSBE. It's a great organisation for any keen botanist. And once you join, you'll have access to a range of benefits. So this includes things like field meetings, where you'll be in a mixed group with some experienced people, some less so. And it's a great way to learn your plants, grasses included. Highly recommended. Top tip is membership also gives you access to the referee network. So these are specialists in their field, and they very happily accept specimens from you, and they will confirm or otherwise your identification. There'll be all sorts of hints and tips they can give you as well. They quite often point out why you've gone wrong and where to look, but always send them something with your identification. Don't just send them a blank sample, because that tends to infuriate us a bit. Have a go, see what you get to. And it also does a lot of plant recording. So this information is all gathered by volunteers, and the data that comes from that informs conservation policy directly in the UK. So it's a really important aspect of what botanists can be doing. And you can all contribute to this. So you can go out and make plant records and feed them into the system. Start your own herbarium. Again, great little tip. So when you've got a plant and you've got the identity correct, you can press it. And again, we'll put a link in to show you how to do this, and make yourself some herbarium sheets. Now, like when you're recording a plant, it's good to get into the habit of putting these 4 key pieces of information. So what is it? In this case, mat grass, of course, as any other example. Where did you find it? So a grid reference, preferably from a GPS or something, but also a description of where you found it. So in this case, in the New Forest in Hampshire, so that just means that if I did get the grid reference wrong, which happens, somebody's got another thing they can latch onto to try and pin the location. When, again, really important, when did you find your specimen and who are you? So that in future, if someone's leafing through records, they can say, yeah, okay, Matt, oh no, he didn't do his grades very well at all. I'm not going to trust his identifications. Whereas if they looked at my conifer identifications, they'd be really impressed. Really useful tip. Start your own herbarium. Just get things on paper and keep them in plastic sleeves in a folder or something. And that means you've always got something you can refer to and compare when you find other grasses. Most important of all, just get out and play, have fun, practice. Be prepared to get it wrong. Getting it wrong, strangely, means you actually know more than you think you do, because you'll know when you're wrong. And it's not a problem. People get it wrong regularly, and each time you're going to learn a bit more. So even after today, go out this afternoon, go out tomorrow, see what you can find.